we're going to talk about a molecular study aimed at understanding how to stop the COVID epidemic. This is something we'll talk about with the next presentation with Lidia Pieri, Sybilla Biotec. Good afternoon.
Good afternoon to you and to all the people watching us. First of all, where are you now? I'm at home. I live in Verona. You know that we have a lot of people connected from all over the world. We do have translation into English, of course. Now, you told me something about your study, and your work is a very practical contribution. So I would like you to describe your project. I think you have a presentation. Yes, my presentation is in English, and so this is, of course, in order to help people watching us from abroad. Well, you can decide speak English or Italian. It's up to you, of course. Would you prefer to speak in English?
It's okay.
Yes, okay.
So can you see my screen?
Yes. Okay.
Great.
Now it's okay.
So can I start? Great. So the question is it possible to combine new biotechnologies and modern supercomputers to find a cure for COVID-19? This would be really great. At Sybilla Biotec, we have a cutting-edge new drug discovery technology, and we would like to use it. And the INFN, that is the Italian National Institute for Nuclear Physics, has let us exploit a significant amount of the supercomputers which were used to discover the Higgs boson at CERN, from Higgs boson to biology. Well, this allows us finally to work on COVID disease. At Sybilla Biotec, we discover molecules that can become new drugs. Now, in general, drug discovery is based on finding small molecules which impair the biological activity of specific proteins. There is a problem somehow. Some proteins are apparently undruggable. In Sybilla, we try to drug them because we act in a completely different way. We use an innovative drug discovery protocol that has been developed by our founders. In particular, we use computers and protein folding simulations. Let's see what it means. So what are proteins? Proteins are the molecular nanomachines that perform most of the biological function in cells. All the info about the chemical structure of a protein is encoded in the gene and written down in the RNA. Then the ribosome prints this information in the form of a linear chain of amino acids. At this point, the protein folds and reaches the final conformation, which is called the native state. Only once the native state is reached, the protein becomes biologically active. And traditional pharmacology acts here on the native state to block the biological function of the protein. Instead, we act before, during the folding process. In particular, we stop the folding process in order to suppress the protein expression in cell. How can we do that? Well, during the protein folding, the system spends a lot of time in intermediate states. So we can identify molecules that bind to these intermediate states and interrupt the folding process. So what happens? The protein doesn't get to the native state, and the cell recognizes a problem in this partially unfolded protein and destroys it through a quality control mechanism. This is why our technology is called PPI-FIT, that is to say, pharmacological protein inactivation by folding intermediate targeting. So we don't allow the protein to get its biological function. Now note, targeting an intermediate state could not be done before. Why? Because in order to do that, you have to perform a protein folding simulation at atomic resolution. The standard computational methods would take much more than a human life to simulate the folding of biologically relevant proteins. However, our scientists have developed a more powerful approach which exploits mathematical methods taken from theoretical physics. And this allows us to perform this protein folding simulation with atomic resolution. I think that this innovation is really a beautiful example of how fundamental research can improve the real world. And this in particular was carried out by a researcher of the Italian National Institute for Nuclear Physics. That's why we have this link, which was so important with INFN. Now, our technology has been tested using our new protocol for drug discovery, PPI-FIT. We have actually been able to find molecules which could suppress the expression of different proteins in cells. Look at this example. These are experimental data and show that when the cell is exposed to the molecules that we find with our approach, the expression level of the target protein is reduced. Proteins are disappearing inside the cell. Now, let's understand how Sybilla can help in find a therapy against COVID. Everyone sadly knows this graph. We want to target a protein which is called ACE2. This protein is the cellular receptor of the virus. That is to say, the key that the coronavirus uses to enter the cell and start the viral replication cycle. Now, if we decrease the level, the number of ACE2 in cells, this would reduce the infectious power of the virus. But more important and more generally, ACE2 was reported to be the cellular receptor of both SARS-CoV-1, the virus that caused the epidemic in 2002, and the present SARS-CoV-2. This means that a compound found which is capable of reducing its expression in cell could be useful now, of course, but also possibly in the case of future coronavirus pandemics. This is our pipeline. It consists of 4 steps. First, we have to characterize the protein folding pathway of ACE2. This requires a very large amount of computing time, and we could significantly speed up the simulation as INFN has given us access to their computational infrastructure. After we characterize the druggable pocket on the intermediate state of this protein, we will run a drug screening campaign to find a potential drug. And then we go on in wet lab. We go to the lab in order to test the effect of such molecules in vitro. To test this effect both as far as the expression of ACE2 is concerned as in the case of its effect on the virus. Now look at this movie. It's beautiful, isn't it? This is the preliminary result showing one folding pathway of ACE2. Note that actually the chain spends time in a few intermediate configurations before getting to the native state. We are using 3,000 computing cores of INFN, and in 2 weeks of calculations, we will produce plenty of simulations like this to achieve statistical significance. Here is an example of a well-structured intermediate state that we have already found. And actually, we are very close to the end of the first step of our pipeline because we will be able to release and publish the results on the intermediate state soon after Easter. And then these results will be made available to the scientific community because we hope that everybody that can help will work on this. So this is the people that makes it possible inside Sybilla, founders and workers. All of them are very important. So I think I can stop.
Okay. Lidia.
Sì? Ti sento ma non riesco a togliere la condivisione dello schermo.
Non ti preoccupare, va benissimo. Va benissimo.
Okay. So the main goal is to block, to stop the virus before it enters the cells, to make sure it doesn't have the key to enter into the cells. And you're saying this is important for the SARS-CoV-2, but could be important also for other viruses. Yes, because such a cell receptor is common to SARS-CoV-1 and SARS-CoV-2, so the pandemic in 2002 and the current pandemic caused by SARS-CoV-2. Of course, we hope there won't be a third epidemic, but if this happens, it is likely that the virus is going to use the same receptor. We hope we have a treatment for COVID-19, and we hope to have a drug soon. And if we manage to do that, we will be ready to face other pandemics caused by SARS-like viruses. Now, it is quite clear that scientific research has been supported by also the technological instruments. You have computing instruments which are so important, and this is very important. So scientific research on one side and technologies on the other. Yes, it was a very fast response, a synergy together with INFN, and we started from the research, basic research, and with the basic research, well, you think about so many things, but then when you have the technological transfer, which comes from basic research, well, this is important because you can use your creativity, the talent of researchers, and then you can create a business line. And when the emergency started, we at Sybilla, we decided to invest our resources in ACE2 folding, but with our resources, that process would have been too long. So we contacted the INFN, and they made available to us computational power that was really impressive. I've been working at CERN so many years ago, and these computers actually were made available to us, and we are just a startup, but this actually confirms that everybody's trying to collaborate. It is a very strong collaboration. So collaboration between general practitioners, but also the decision makers of our cities and towns, and so the collaboration between them would be also important. So social innovation as well could be involved, and we hope all the best for the future. Thank you so much for describing your project, and I wish you all the best. Thank you.